Hello guys, I am Shubham from Food Tutorials. This video is about mismatch repair. So before we start, subscribe to our channel and press the bell icon to get notified whenever new video is out. So let's get started with the video. In the previous video, we have discussed why there is need to repair DNA. What are the DNA damages? What are the errors that we have discussed in the previous video. From this video, we are starting with actual repair pathways. So this is the first pathway, that is mismatch repair. So this is for the, we can understand from the name only, mismatch. Okay, mismatch. Mismatch means what? Incorrect nucleotides are added, right, during the DNA replication. Okay, so this is known as replication error, right? This occurs due to result of replication error, which escapes proofreading, right? So proofreading is escaped, and thus the errors get fixed. Incorrect nucleotide get fixed in the DNA. So this incorrect, or we can say mismatch pairing, has to be repaired, right? So here comes this repair pathway. So this is also for the increasing the accuracy of DNA replication. So this is just an example. We can see here, this is a parental strands, right? First round replication incorporates incorrect nucleotides, right? Misincorporation. So this results in C, A and G. It should be T, right? Here it is T. It should be T here, but instead of T, G is there, which results in incorrect pairing, right? So there is no pairing basically, incorrect nucleotides, and this known as distortion. Okay, so distortion is present, and we can see this is correct one. See, A and T. One strand is correct, one strand is incorrect. Now, in this, after first round, the repair has to be done. Okay, a repair has to be done in the first round. If repair is not done, then second round application takes place. Alright, second round application takes place, and here also we can see one strand is having AT, right? Because of this AT, and here it is CG. So this results in the mutation. This is known as mutation, and this will get fixed. So this has to be corrected in the first, after first round of application only, right? So let's see the pathway now. See, this is the first protein known as mute S. So mute S will scan the DNA, okay, and recognize the mismatch. It is having a structure like this. We can see in this picture, when the distortion is found. So this is a distortion, known as distortion, right? Incorrect nucleotide. So when distortion is found, see, the structural changes happen, right? Structural changes occur in the mute S. See, we can see this is holding the incorrect nucleotides, right? Distortion. So this mute S is a ATPase. ATP will come and get bind to this proteins. Then there will be a hydrolysis of this ATP. This is a ATPase enzyme. So ATP has bound to it, and then it recruits other protein, mute L. Okay, mute L, and the mute L will again recruit another protein known as mute H. So here the ATP hydrolysis has occurred, right? Then this mute H will do the nick, basically cut. Okay, cut near the mismatch, right? Cut near the mismatch. Then will come helicase. Okay, helicase, UVRD. In the picture, the helicase is not shown, but helicase is for we know to unwind the DNA, right? DNA is double stranded. To cut the strands, helicase is there to unwind the structure. And then the exonuclease will come. It will digest the strand. You can see here, there is a gap. Okay, till the mismatch nucleotide is there. Okay, so there will be gap, and this gap will be filled by DNA polymerase 3. This we are seeing in E. coli, right? This we are seeing in E. coli. So we can see here the mismatch has been repaired, right, with the correct nucleotide. So let's take a summary. So this is in E. coli. First enzyme, mute S. From S, we can keep in mind scans and recognizes the mismatch. Or we can say distortion. Second protein is the mute L. Mute L, right? And then this will again recruit a third protein, mute H. So mute S is a ATPase. You have to remember this. ATPase means it will bring ATP hydrolysis. And mute H is endonuclease. Endonuclease. We know what is endonuclease. It makes a cut in the DNA. Here we can see cut has been made. Mute H introduces nick, right? Nick. And then comes exonuclease, right? So these three are the important proteins, basically mute for the mismatch. Okay, we can keep in mind like this mismatch, right? Mismatch means we can say this is one type of mutation, and this has been corrected by this mute proteins, mute S, mute L, and mute H. Basically, this comes in the mismatch repair. And then comes exonuclease. Exonuclease will make a gap, right? Exonuclease making gap, or we can say digest, or digest the strand with incorrect nucleotide with an incorrect nucleotide. And then comes DNA polymerase 3, which we know it is for the replication, right? It will incorporate correct nucleotide. Okay. So basically, which exonuclease is being used? So which exonuclease is used? There is also a types in exonuclease, right? We will see that if the cut is in the 5 prime side, see this is a 5 prime, right? And this is a 3 prime. Okay. If the cut is in the 5 prime, we can say nick. Okay. So these types exonuclease come, exonuclease 7 or rec-J, which will digest the DNA in 5 prime to 3 prime direction, right? Exonuclease 7 or rec-J. Okay. And in case of 3 prime cut, exonuclease 1 will come. Exonuclease 1, which will digest the strand in 3 prime to 5 prime direction. Now, what is this? What are these areas which we can see? So these are the methyl groups. We have discussed this previously also, that how does this repair system recognize the parental strand and the daughter strand? So the answer is methylation, right? So newly synthesized strand is unmethylated and the parental strands are methylated. So these are the methyl groups. These are the methyl groups. So we can see parental strands are methylated and daughter strands are, or we can say newly synthesized strands are without methylation. So thus the repair system recognize the daughter strands and act on it. Alright. So this is all about mismatch repair. So that's it for this video. I hope this video helps you. If you find this video helpful, do let us know in the comment section.